Hello everyone, welcome to the Immunology Capstone Review. I'm going to start with telling you a little bit about what you can expect today and this morning as we go through these. First, we're going to start off with part 1, which is going to go over about the first third of everything that you learned in immunology while you're at RAS. At the end of the first section, you're going to have a case study to work on. I'm going to leave you with the case study, which is included in the end of your PowerPoint, and it's put in with that whole grouping. It says Immunology Capstone Case Study Number 1. So as we conclude part 1, you will have time to stop and work with your classmates to figure out what you think the solution is and to answer the questions at the end of the case study. When you're done with that, you can begin working on part 2. At the beginning of part 2, I will go over the answers to the questions on that case study at the beginning of part 2. So after that, we'll go on ahead and we'll go over part 2. At the end of part 2, you're going to have case study number 2. Again, when you're done with that, you'll have time in between to work on the case study, try and figure out the answers to the questions, and at the beginning of part 3, I'll review those questions. At the end of part 3, you'll have a third and final case study, and with that case study, I will go over that at the beginning of our live question and answer section. So you'll have a total of 3 case studies to work on, along with the review information that that you have been given. So what you're going to see today is going to follow the PowerPoint handouts that you have that were given to you by your program coordinator. If you don't have those, make sure you pause this and get those from your program coordinator. All right, let's get started. We're going to start going back in the past to the beginning of immunology. We see Louis Pasteur, but he was in the 1880s. Even prior to that, there was some pretty crazy things that we used to do to try and create immunity in people. I don't know if you remember, even the first couple paragraphs of your immunology textbook, it talks about in 100 AD, the Chinese used to take the smallpox virus lesions that were on people's skin. They kind of turn into scabs, kind of like chickenpox would turn into a scab. They would pick the scabs off, crush them up, and have people inhale those dried up pieces. Now, what that would do is invoke an immune response within the body where you would make antibodies to those antigens, which were essentially attenuated or even dead. So they didn't really cause infection within the host. But the antigens were still there, so the bodies are still able to make an antibody to the smallpox virus. Well, that's desirable, but I would not call inhaling the crust of someone else's scabs desirable whatsoever. So good thing as we moved forward, they came up with other ways to come up with vaccines and things. Louis Pasteur, we call the father of immunology. He made some major changes that obviously did not involve inhaling the other people's scabs. It involved making actual vaccines to things like smallpox and anthrax and things. So what is the difference between immunology, the immune system, and immunity? Immunology is the study of the way the body protects itself from non-self. Now, this can be, it can be desirable effects where you're able to fight off pathogens or undesirable effects of immunity such as autoimmunity. All of this involves your immune system. Your immune system is specifically the part of you that helps protect itself from non-self. Once you have protected yourself from non-self by way of making antibodies and having a memory-type response, you have developed immunity. So what are the body's first lines of defense? A little bit of review here. Unbroken skin. So skin is a great line of defense as long as it's not broken. Tears, saliva, mucus, things like that. There are a couple different types of defenses involved. We've got the natural immunity and the adaptive or acquired immunity. Things that you are born with. The second you're born, you are protected by a cellular and humoral components. The cellular components involve mast cells, neutrophils, and macrophages. Those are your cells that are capable of phagocytosis. They're able to eat foreign organisms and just ingest them and kill them without having a whole lot of prior exposure to them. The humoral natural immunity refers to the chemical type portion that helps out. Complement, which I'll show you guys. Remember the complement cascade? No fear, I will go over that in part 2. Lysozyme, which comes from tears and saliva. And interferon. Interferon helps fight off virally infected cells. Then we have the adaptive immunity. So after you're born, and your mom takes you home, and then you've got Aunt Ethel and Grandma and everybody sitting around the living room, passing around the new baby. They're breathing on baby, exposing the baby to all kinds of things that this baby has never seen before. So the baby needs to adapt to that new environment. That's the adaptive immunity. The cellular components of this include T cells and B cells, which are involved with memory. Once you've seen it before, your body's familiar with it, those cells come in and help out. B cells, those are the ones that are really good at secreting immunoglobulin when foreign organisms do invade. And then we have the humoral part of the adaptive immunity. This is where the antibodies are made to help fight off those foreign bodies and things. We also have cytokines, which are the chemical messengers that cause the talking between the cells so they can communicate and everybody in the immune system knows what's going on, that there is something that needs to be fended off. So let's look at a couple different types of that acquired immunity. We can have active immunity or passive immunity. Active immunity, two examples are active natural and active artificial. So what's the difference? If you have active natural infection, that would be me when I was little. I got the chickenpox virus. My cousin had chickenpox and gave my mom a call. So what does my mom do? My mom runs me over to my aunt's house. So I go play with my little cousin. We kiss each other and hug each other to make sure that I gave my cousin the chickenpox. Oh, that's silly, you ask. Why would you do that? Well, back when there wasn't a chickenpox vaccine, that was the common way to get chickenpox. And another thing is, if you don't get chickenpox when you're little and you get it when you're older, or if you're immunocompromised, it can become almost deadly. So when you get it when you're little, it just ends up being some itchy scabs or a rash all over your body. And it's nowhere near as big a deal as it is if you were older. So that would be the active way to get an infection, active natural. Active artificial would be, nowadays, my kids got a chickenpox vaccine, also called the varicella-zoster vaccine. So that is the difference between active natural and active artificial. Then we've got the passive immunity. We can have passive natural and passive artificial. Passive natural is the placenta and colostrum. Antibodies are able to cross the placenta, especially IgG antibodies, those are the only ones that do, cross the placenta and help protect the baby until the baby's born, then they've got to fend for themselves. And also colostrum, which is the breast milk that the babies get right initially after they're born. The other way is passive artificial. This is the injection of an immunoglobulin directly into the person. This is would be an example of someone who does not have their own immunoglobulins, or if some people that are exposed to a hepatitis B virus by way of a needle stick, sometimes they give them the hepatitis B immunoglobulin. So that immunoglobulin goes right into that person and starts fighting off that hepatitis if it is there. It's short-lived. It's not something that's constantly being excreted in the human body like a B cell would create. But if they can give them some injections of that, it might prevent a hepatitis B infection from occurring in the first place. All right, a couple other types of reactions we have. Hypersensitivity reactions, which would be kind of like a bee sting, refers to an anaphylactic reaction, which involves IgE. We're going to talk about that in more detail later. PAMPs, pathogen-associated molecular patterns, also called toll-like receptors, are an important part of immunity. Those are what tell your body that it belongs to it tells your body that an organism is foreign. Some organisms have the exact same molecular pattern on them, so your body is able to recognize, even if it's never seen it before, that, hey, this guy is foreign, I need to get rid of him. Some other components that you should be familiar with the immune system include mast cells. Those like to release histamine and heparin. Those are what make you all itchy and sneezy when you are allergic to something. Neutrophils are phagocytes. Macrophages are also some types of phagocytes. Complement obviously comes in and does the final job of destroying a foreign organism. Lysozyme is the chemical component of your tears and saliva that when it helps protect you from foreign organisms that way. Interferon is the way you protect yourself from viruses. Antibodies, which are secreted from B cells, can tag foreign bodies so that our immune system is prepared to destroy it. and cytokines, those chemical messengers between the cells. Notice I'm kind of bringing up some of these things a few times, probably because they're pretty important. All right. You notice I have chapter 2. I did divide this up as it goes through your textbook. So kind of taking you on a quick review of the entire textbook. So we're moving on to the second section here. So what is an antigen? An antigen is something that makes a organism itself. So all things have antigens on them. Cells have antigens on them. Bacteria have antigens on them. Those antigens have an epitope or an antigenic determinant where an antibody fits into it like a lock and key. Now those antibodies are able to kind of flex their arms a little bit and fit where they need to fit. But be familiar with the fact that the antibody fits onto the antigen of the foreign body, and the epitope or the antigenic determinant, they're the same name, is where the antibody would attach to. Our bodies have what's called histocompatibility antigens or autoantigens that make our body aware that it's self. Now, obviously, we want our immune system to know if a staph organism comes in and it destroys it. But what about our own cells inside our body? We have our own major histocompatibility to make sure that our body knows it's itself and it doesn't destroy itself. Now, as we'll talk about autoimmunity later, sometimes those types of antigens become altered and our body does start to destroy them. Or we do start to make antibodies to our own cells, which is an unfortunate part of immunity. Well, what is an antibody? An antibody is there to help tag an organism that is foreign so our body knows to destroy it. It's also called an immunoglobulin. Same thing. We do have 5 classes of immunoglobulins. We got the IgG, GM, IgD, IgE, and IgA. Some of the things that you need to know about those, immunoglobulin IgG is in the largest concentration in your blood. He's involved in the primary response, but he's largely involved in the secondary immune response, which we're going to get to at the bottom here in a minute. He's also capable of crossing the placenta. IgM, he's that big pentamer. He's huge. He's involved. We use him in a lot of our blood bank reagents too because he's so big and wonderful to use. But he comes in as a primary response. He can come in and take care of the job very quickly. Plays a large role in the primary immune response the first time you're exposed to a foreign organism. But the second time you're exposed to the organism, IgM doesn't play as big a role as IgG does. So IgG plays a big role in secondary, IgM plays a big role in the primary response. IgD, he's found on the surface of B cells, but he's not really in a lot of, he's in the smallest concentration. You don't find a whole lot of him floating around. IgE is the next one. He's involved in the hypersensitivity, the allergic reactions. He helps release heparin and histamine from basal cells. So that's what makes you all itchy and sneezy. And lastly, we have IgA. IgA is secreted in tears and saliva and help protect you that way. All right, so next we'll move on to the actual antibody itself. I'm going to kind of skip down a couple lines here. And you can see I have down on the bottom a picture of an antibody. They look kind of like a Y, okay, an immunoglobulin. You need to know that the N-terminal end the N-terminal end is known as a variable region and binds to the antigen. So right here would be the Fab region that binds to the antigen. And then down at the bottom we have the FC region over here. Also what we have are the different types of chains, and I'm going to have to draw a new one for you. We've got the heavy chains, which are the longer ones right here. This would be your heavy chains. Pardon my drawing, it's not easy to draw with the mouse. And then we've got your light chains on the outside. Okay. We also have, and I'll draw circles here. We have our variable region, and we have our constant region. So the antibody class and subclass are determined by the constant region of the heavy chain. Okay, so that would be our second one that I drew over here. Apologize for my drawing, it's terrible. We have different types of bonds that are included in antibody. We've got van der Waals forces, hydrophobic bonds, hydrophilic bonds, all kinds of bonds involved in there. I have on here be able to draw and label an immunoglobulin. Well, you would not have to draw an immunoglobulin for your post-capstone test or for your boards, but be familiar with the different pieces of it. What the part that binds to the antibody would be the Fab region, then you've got the FC region on the other side. Know and draw the phases of the primary antibody response. Again, you don't have to draw it, but be familiar with it. I do have the primary and secondary responses over here on the right-hand side. And this first lump that you see here, the red one, is your primary antibody response. The IgM comes in a little bit, but then IgG kicks in really, really high. So actually you could call this whole thing the primary antibody response. You've got the lag phase, a log phase, a plateau phase, and a decline phase. So they are all, actually the red one is the primary and the blue one would be the secondary response. IgM plays a bigger role in the primary, IgG plays a bigger role in the secondary. The secondary response is also called the amnestic response. All right, so what is a titer? A titer is the reciprocal of the largest dilution that we find a positive result. So a titer of, let's see, if you had a titer of 1 or of 8, that would be the same as this dilution. Okay. So if you ran a titer and you had all of your tubes had agglutination in them, including the 1 to 8 dilution, but the 1 to 16 was negative, you would report that as an 8. Or it was positive in the 1 to 8 dilution tube. So what is so great about monoclonal antibodies? This is our next thing on here. The monoclonal antibodies are antibodies that are cloned from a single cell line. So they're all very, very specific. We use those a lot in laboratory testing. All right, moving on to different types of agglutination methods. We do a lot of agglutination methods in the immunology serology department, so we tend to cover it a lot more here. So different principles behind precipitation, agglutination, co-agglutination, for example. Precipitation, a type of precipitation is flocculation. This is the binding of a soluble antibody with a soluble antigen that creates an agglutination reaction. It kind of looks like a fine particulate agglutination on your slide. Examples of this are the VDRL and RPR. VDRL is usually done for syphilis on spinal fluid, and the RPR is a rapid plasma reagin, which is a protein that we test for when a patient has syphilis, kind of a screening test. But those are two popular flocculation tests. Then we have agglutination. Agglutination uses latex or colloidal charcoal as a carrier. This one's quite common. I'm sure all of you have done this in some way, shape, or form, either in your labs or at your clinical site. We have hemagglutination. This is where it's attached to a red blood cell. And we can have direct bacterial agglutination, where we have antibodies to the actual bacteria itself, and the bacteria causes the agglutination. There's also co-agglutination, which uses other types of particles to cause agglutination. Be familiar with what zeta potential means. That is the degree of repulsion between cells. You see this little electron-proton cloud that I have over here on the right-hand side. You'll see lots of positive charges and negative charges around. And what the general negative charge between cells does is it causes the cells to repel against each other so that they don't stick together. I mean, it would be very unfortunate to have all kinds of cells traveling through your bloodstream and sticking together, right? So you have zeta potential that kind of keeps them repulsed away from each other so they don't stick together and they can flow freely through your bloodstream. But that can cause a problem with agglutination, right? If there's agglutination that's going to happen, we want it to happen. We don't want zeta potential interfering. So there are some things that we do. The two stages of agglutination, and we'll get to how that works, is sensitization and lattice formation. Sensitization gets things started, and the lattice formation is actually the combining of antigens and antibodies in a big network, so you can see the agglutination. How do we enhance agglutination? We can centrifuge it. That overcomes zeta potential by forcing the cells together. Enzyme treatment strips the zeta potential off the cells. Colloids can do something similar, and the anti-human globulin is another way that we can enhance agglutination. We can have issues can be caused with agglutination when we have pseudoagglutination. This is something you've probably heard more about or may have seen in your blood bank rotations. This is caused by something called rouleau. And when you have rouleau, there's such an excess amount of protein. It can be caused by multiple myeloma, Waldenstrom's macroglobulinemia, things that have extra protein in the blood, and it can kind of strip that zeta potential off as well and cause those cells to stick together, and they look like stacked coins. So it can look like agglutination to the untrained eye. Another example, pregnancy testing sometimes uses antibodies to the beta subunit of HCG. And I remember talking about this in this chapter, which is why I kind of added it here. It uses like an ELISA type of method with looking for the beta subunit of HCG. The last thing on here is, what is agglutination inhibition? That is the presence of, and it's going to have negative test means the test is positive. No agglutination equals a positive test. Here's an example. I'll use the HCG as another example, and this is from your textbook. The incubation of the patient's specimen with anti-HCG, then you add latex particles coated with HCG. If HCG is present, it neutralizes the antibody and there is no agglutination. If no HCG is present, agglutination occurs. So it's opposite of what you would think. Agglutination equals a negative test result. So if you're taking your boards and they said there was agglutination with an agglutination inhibition, then you would have to put that the test is negative. Okay? It's opposite of what you would think. And that's a real possibility that you could get on your boards. All right, now we're going to start moving on to some cells. We're going to end this part with the granulocytes and monocytes, and we'll start the next part too with the lymphocytes and things. So let's move on to the granulocytes and monocytes. First, you need to know how the development of blood cells occurs. First, it starts in the yolk sac of the fetal yolk sac. Then it moves on to the liver and the spleen as the baby's maturing, and then finally to the bone marrow. Different types of cells in the granulocytic series include neutrophils, which are very efficient at phagocytosis. It's your major defense against bacteria and fungus. Then we have the eosinophils. They're regulators of inflammation. They have defense against parasites. They're very good at that. Basophils are the next one. We don't see those guys very often. They're involved in hypersensitivity reactions, and they release histamine and heparin. So there's that itchy, sneezy, etc. IgE is usually involved in that as well. So what is this process of phagocytosis? Well, phagocytosis is the actual eating of, or the ingestion, I shouldn't say eating because they don't have teeth, the actual ingestion of a type of bacteria. The first thing that happens, and I call it can an elephant dig dirt, CAEDD, chemotaxis. That is the chemical attraction of phagocytosis towards a foreign particle. It's kind of like there's a chemical that's released and it's saying, okay, come here, there's a foreign particle present. Then it adheres. It's got the adherence. You can see it adhering in this picture over here, particle. Then we have engulfment. In engulfment here, it starts to make a phagolysosome in here. So we have this whole thing start to happen. We get the lysosome and we get the particle. They come together, make a phagolysosome, etc. Then there's digestion that occurs within the cell, starts to destroy it, and then destruction. It becomes destroyed. Normal flora, it's important to make note of at this point that your body is familiar with normal flora. It's bacteria that keeps us healthy. So your body does not do this to normal flora. So if you ever see the word normal flora come up, that's what it refers to. We just discussed what a phagolysosome was. That is the fusion of the phagocyte, or the particle that was phagocytized with the lysosome, which starts the destruction process. Next couple types of cells, monocytes and macrophages. Well, a macrophage ingests foreign particles and infectious organisms by phagocytosis as well. We call him the garbage truck of the blood in the blood. He can make through tissues. He can, and depending on where he's located, he gets a different name. Kind of like if he's in the tissues, he's a histiocyte, or if he's in the blood, he's a macrophage. So he can end up in all different ways and have different types of names. But he's really good at phagocytosis. He also induces an immune response and secretes some biologically active molecules as well, as you can see I have on there. Then we can also have acute inflammation. What happens with acute inflammation, when that occurs, diapedesis happens. And these macrocytes, monocytes, macrophages, and granulocytes are really good at diapedesis, which is the movement through a vessel wall into the site of inflammation into the tissue. Another thing that can happen is sepsis. Sepsis occurs when there's an overwhelming bacterial infection in the body that has entered the bloodstream. We find extreme temperatures. We find, you can even have seizures. It just depends on how overwhelming the infection is. But that is an overwhelming infection in your entire body. That's the definition of sepsis. So let's go into a couple types of diseases. First, neutrophil disorders is what is the difference between a qualitative and a quantitative neutrophil disorder? Hopefully by now you guys have the words quantitative and qualitative down pat, but I'll give you a little review just in case you don't. Let's say you have a neutrophil. If he had a qualitative defect, there's something wrong with the quality of that cell. He is not able to kill off bacteria like he should. He's poor quality. Now, if you have a quantitative defect in neutrophils, that simply means you don't have enough neutrophils and the quantity is not correct. So that's difference between qualitative and quantitative. First disease with neutrophils, Shinya-Higashi. That's kind of a fun one to say. Here they have an issue with giant granules and they have impaired chemotaxis. While these giant granules may have myeloperoxidase and things in them, they are not really good at moving towards the area they need to move to destroy things. So they have impaired chemotaxis. They really can't destroy things like they should. The next one, chronic granulomatous disease, not sometimes called CGD. This one is where you have issues with the cascade of events required for hydrogen peroxide production. Hydrogen peroxide is present in the cell to do the final destruction of a bacteria. If you don't have that capability within a cell, you're not going to be able to fight off a whole lot of anything. The next ones are monocyte macrophage disorders, Gaucher's disease. These people have a deficiency of beta-glucocerebrosidase enzyme, which is involved in the killing of microorganisms. So you get overwhelming infections pretty much in all of these disorders. The next one is Niemann-Pick disease, and that one has a deficiency of sphingomyelinase enzyme. And my students always laugh at me because I'm like, Niemann-Pick disease, pick, and sphingomyelinase sounds like finger. So you pick your nose with your finger. So that's how I memorize that one. And the last one is leukocyte adhesion deficiency. This is one where the cells just have a hard time adhering to the side of a foreign organism. And that when that happens, you have a really hard time healing. So, for example, babies, one of the ways they find this is that a baby's umbilical cord just doesn't fall off. And if any of you have had a baby, you know how quickly that happens. Like within a week or so, that thing turns all crusty and it falls off. It's kind of gross and icky. But if there's leukocyte adhesion deficiency, it doesn't stick really well, and it has a hard time, and that umbilical cord just does not fall off. So that's one of the first signs that they find with that. All right. That was a quick and easy part one. I hope that wasn't too painful for you. So what I want you to do is look at the next couple pages of your packet. In that packet, you will see case study number 1. Your case study number 1 is about a 7-year-old girl from Pakistan. She's got recurrent infections, and as you can see, she's got, it's kind of hard to point out, I'll point this out to you. This is the actual picture of her. She's got this top right-hand side, she's got blepharitis. She's got infections in her eyes. Her gums are extremely puffy, extreme gingivitis. Some of the other pictures were the crust behind her ears. She had bald spots on her head where there was big sores that weren't healing. So go through and read that whole case study, and you can use your book. You can look back through the PowerPoints, whatever it is. You can use the internet even to see what you think might be the cause. And I'll give you a hint. It is something that we have talked about in the last 4 slides. So see if you can work as a group to answer the 2 questions at the end. And the 2 questions are, which lab results are abnormal? So I want you to go through all those and highlight those. And which disorder is likely present? So we'll cover that at the beginning of the next segment. Go ahead and take some time. Take a half hour or so. Try and figure this out. Take a soda break, whatever you need. So I will see you shortly.